Welcome back. To determine evolutionary relationships, scientists like hardcore detectives collect all sorts of clues, both morphologic and genetic, to then piece together how the different organisms might be related. So we have two big categories of evidence we use. We have morphologic, how things look and function, and then we have genetic. We have two ways to measure evolutionary similarities. We have both morphology, form and function, and genetic evidence that is used to make those connections between organisms. When an organism's traits match up both physically and genetically, we call them homologous structures, the ones that share the same evolutionary root. So homologous structures have similar features in different species. They are inherited from a common ancestor and they're displaying divergent evolution this way. Remember the awesome examples from last week? We talked about how the bony forearm structure of humans, dogs, birds, and whales look the same. Let's add a few more species. Bat wings and horse forelegs. See, look at the bat wings and the horse forelegs. And then these are the ones we already looked at. We got the birds and the whales. They all have the same groupings of bones than the humans down in here. They might not look exactly alike at first glance, but if you look under the hood, there's a familiar set of bones just arranged in different ways. Nature didn't just take one bone, but a grouping of several that work well together and arranged in similar ways, even though they can end up with unique functions. A bird flies, or a bat can fly as well, a horse trots, and a whale pushes water away to swim. But beware of appearances. Tiny genetic tweaks can lead to big differences in how creatures look on the outside. That's why chimps and humans can be 99% genetically similar, yet show considerable anatomical differences, like the jaw that's protruding here in a different way. And let's not forget, we walk upright while our chimp cousins swing through trees. On the flip side, unrelated species can look strikingly similar. They evolve under the same environmental pressures like fish and whales, who both have sleek bodies and fins for swimming. Or we've also seen the example of the Arctic fox and ptarmigan, both having white covers in the winter. These look-alike features are results of convergent evolution. Here, similar traits evolve independently. We call them analogous structures because they develop separately to handle the same challenges like moving in water or not being seen by a predator, for example, but they do not come from the same ancestor. Another great examples we've mentioned before for analogous structures are the wings of insects as compared to the birds or bats. See here? We got the wings of insects and then we got the birds and the bats. The birds and the bats come from our regular skeleton, whereas the insect stems from the exoskeleton, a completely different ancestral lineage. This is where scientists have to sort out the real family trees from the red herrings. Similar features can be homologous, sharing a legit evolutionary origin, or analogous, just a copycat solution because of environmental demands. Disentangling the two can be pretty tricky. Thanks to modern molecular systematics, we can compare genetic sequences that figure out who's truly related to whom. The logic is simple. The more DNA or amino acid sequences in common, the more closely related the organisms probably are. And yes, it can get complicated like those little fruit flies that share a surprising chunk of DNA with us humans, for example. Another cool aspect of phylogenetic analysis is what it tells us about disease spread and conservation. On the disease side, researchers traced the journey of the antibiotic-resistant MRSA strain as it hopped around the globe. MRSA stands for methicillin-resistant Staphylococcus aureus, if you want to impress a trivia night. Turns out it might have spread from very few patient zero introductions instead of popping up everywhere independently. This means public health folks can focus on isolating those first carriers and stop the spreading in the first place. In conservation, phylogenetic analysis helps us figure out which species deserve some extra TLC, some tender loving care. Let's say we've got two endangered species. One is part of a big cluster of close relatives, while the other is the last lonely twig on its branch of the evolutionary tree. If that single species goes extinct, we lose an entire unique lineage. Way more evolutionary history vanishes compared to losing one member of a large sibling group. So by checking out the evolutionary roadmap, we can make more strategic decisions about who to protect. To build these roadmaps, we also call them phylogenetic trees or cladograms, biologists use a method called cladistics. What it does, it groups creatures into the clades, which are basically different groups, and everybody in that group is a descendant from a certain ancestor that had a new trait. See this picture here? It shows a clade. See here, this is a clade of all the critters that have amniotic eggs. And that includes humans, rabbits, lizards. All those with amniotic eggs are in that group, that branch of the family. We call a clade also a monophyletic group. See here, mono refers to one, and phyletic is the evolutionary relationship. So cladistics is a method of organizing organisms into groups called clades based on their shared evolved characteristics, using branching diagrams, which we call cladograms, to depict evolutionary relationships. When constructing these trees, scientists go by shared ancestral versus shared derived characters. A shared ancestral character is something every member of a group has. So it doesn't help us to figure out who's more closely related to whom. In our example, these are the ones that have vertebrae. That's the shared ancestral character in this group. On the other hand, a shared derived character pops up only in some members of the group, showing a newer evolutionary twist. These are the species with amniotic eggs, for example, here that we are talking about. It's like little signposts saying, hey, these species over here are a more recent lineage. In our example, again, the amniotic egg species is more recent. So, shared ancestral characteristics are also known as plesiomorphies. And the word plesio means near or close, and morphes refers to form or shape. So these are traits that are present in common ancestors and found in all of its descendants, indicating a shared evolutionary history. In contrast, shared derived characteristics, we also call those synapomorphies. That's a complicated word. And syn refers to together or with, apo is away from, and morph refers back to shape or form. So these are traits that are shared by two or more species that evolve from that common ancestor. So they are together pulling away, and that's what that word means, synapomorphies. Finally, have you ever heard of maximum parsimony? That's the principle of choosing the simplest explanation with the fewest evolutionary twists. In other words, don't assume a trait evolved, disappeared, and re-evolved unless you have to. Computers do much of that heavy lifting because the possibilities can be mind-bogglingly big. So, maximum parsimony is a method of inferring evolutionary relationships by selecting the tree that requires the fewest evolutionary changes to explain the observed data. The word parsimony comes from parsimonia, which is Latin for frugality or sparingness. So that makes sense. As DNA evidence piles up, scientists keep refining the grand tree of life. Did you know people are more closely related to fungi than fungi are to plants? Pretty crazy. Every new discovery fine-tunes our picture of how life on Earth evolved, and it's definitely something to marvel about next time you're checking out the mushrooms at the grocery aisle or something like that. Good job, you guys.